So we often talk about drug discovery as a costly and long process, and it is understood for mainly for a decade or more, and mainly the two answers to these problems exist. One of these is rather strategical, which is based on use of open innovation as opposed to the closed innovation cycle, in which the ideas are not generated only internally in pharma companies, but can pass from inside as well as pass outside to finally to reach to the drugs. And the other approach is rather tactical in its nature. It's based on the it's close to that used in programming world, the use of agile and lead hypothesis testing. When you can fastly test your hypothesis, you can efficiently research the landscape. So let's focus on the latter approach and what theoretical concepts support this approach. Let us step aside for a moment and assume we ask an experienced organic chemist to predict a log p value of an organic molecule when he or she first looks at the molecule, and you wouldn't be surprised that an experienced scientist will predict such log p value with very good precision, because of the functional group based estimation and the properties of the molecules of organic molecules are mainly determined by the presence of functional groups. That is because the intermolecular interactions, that patterns that are pertinent to these groups, do exert these properties. And actually, not only the log p value, but any other physicochemical properties can be predicted. Even more, I can say that chemists think in terms of fragments and design their molecules for that. But what about drug discovery? Is it so easy in this field? And in this field, the fragment-based drug discovery approach has been known for decades, and it mostly consists in assembling a drug-sized molecule from the small fragments with which are very efficient and binding, and with built-in good properties such as ADMET, etc. And yes, okay. And this approach enables one to span an immense chemical space of possible drug-like molecules and produce them in engineering fashion. This highlights the priorities in building a molecule. First of all, you should need to make a build-up of affinity by a certain fragment. Then you can optimize selectivity and met properties with the latter two properties in some exchange of affinity. And okay, that's the old story. You all know about fragment-based drug discovery, but what's new in the field? And we propose you the same another approach, which is based on the same principle, but the direction of thinking is reversed. what if we start from a whole molecule, not the fragment, but a whole molecule, and we would like to assess which are the contribution of each fragment in this molecule to guide other decision and molecule optimization. And this we called fragment-based drug discovery. In short, we split up a molecule into a set of fragments. We estimate separately the interaction of the fragments in precisely the position of these fragments take in a ligand receptor complex, and estimate the share of binding energy using scoring function, but other methods are also applicable here. And then we split the interaction energy for the complex into the contributions of each fragments. So we essentially, this approach has the same roots and principles as the fragment-based drug discovery approach, which was presented above. But we have run into the misconception, and we called it fragment perception hysteresis. If you talk about fragment-based drug discovery, it's okay, it's fine, Q1 papers exist, and it's a honorable approach. But if we just reverse the direction and use essentially the same methods, we run into the resistance in adoption, and some reviewers say that the thermodynamics in very non-linear stop even thinking about this direction. So actually the adoption is not linear, despite the principles are exactly the same. But leaving aside the psychology moments, we would like to analyze the roots of this hysteresis, and we think we have certain answers to this confusion. First of all, we should consider that the probabilities that are considered in these two approaches are different. Whereas in the fragment-based drug discovery, we talk about the probability of binding of a ligand provided a specific receptor, whatever position, because we determine the activity experimentally. Whereas in the reverse fragment-based drug discovery, which is based on in silico estimation of fragment interaction, the position does matter, and it's a precondition for the probability. It is we estimate the probability of binding a ligand provided the specific receptor and in specific position. Having this probability with conditioned, we can apply the Bayes theorem and factor this probability into the components, and we hear that these two components are related by this is a normalizing multiplier, but we think that exactly this term explains most of the confusion. That is based on the probability to find a position of a ligand provided the binding is occurring at a specific receptor. And many readers skip the notion that in reverse fragment-based drug discovery, fragments don't roam over the binding site, but are estimated at their specific position. But we think Okay, this is the main source of the confusion, and we are going to publish our findings here to release this confusion. Okay, that's enough about confusion, and let's talk about the practical applications of the approach. First of all, what we get from the reverse fragment-based drug discovery analysis in silico. We get two types of diagrams. One of which is considers the shares of fragments in the energy and in absolute energy. So we can judge if the fragment is which part of the free energy each fragment accounts for. And the second type of histogram is in terms of ligand efficiency. So there could be a small group, but very efficient, and it's sometimes it's useful to guide decision, as we can see further. So we have spent some time brainstorming what are the main applications of the approach we propose, and we have come up to the two main use cases. First is the rational growth of the ligand, which is very close to the regular fragment-based drug design, and the second is ligand trimming, which provides a fresher perspective. Let's consider an example of the first use case, ligand growth. We reproduced retrospectively the research in which Tankerase 2 inhibitors was sought, and we found that the dedicated fragment shown in blue here could be detected earlier, that it produces the main contribution in binding affinity, and then rational growth could be done using the structure of the receptor, Tankerase in this case. Also, this case shows us that the priority in mentioned above, then you first build up affinity, and then selectivity and admit, also are in place for the final ligand, which was produced by the paper we used for the retrospective analysis. These are the fragment contributions, and we see that the anchor fragments, which gain the main affinity, is in place, and it helped to develop all the series. So let's move to the more interesting case to our feeling. It's the case when you on input have a whole molecule consisting of several fragments, and it is perhaps as results. It is a hit or elite or even elite molecule, which resulted from phenotype screening, and we had such a case when this molecule, which has phenotypic activity, it's lowering in intraocular pressure, and
Dmitry, I'm sorry, half a minute rest.
Okay, I'm finishing. So we could estimate that that fragments 1, 3, and 4 are efficient, whereas the fragments 2 and 5 are not efficient and could and should be eliminated to simplify the structure. So the similar results for the second use case, all results are published. And okay, so
Okay, unfortunately, the time is over. I see two questions in the chat from Dmitry Solodkin and Daria Tsiba, but we have no time to answer your to listen your answers. So I suppose that you communicate with colleagues directly via chat or email. Thank you